I think if we think about the end product of drug discovery and what we're aiming for, we're aiming to create medicines. And of course, for those medicines to be marketed, they need to be effective. And they need to be effective in a human being, in a patient. And so cells as models are a means to getting closer to the patient. The better models we can make, the closer we can get to the disease state and to the patient, and ultimately the greater success we're likely to have, simply because we're able to measure things which are more relevant to those disease states. I think in terms of cells as models, on a very simple level, cells of course are a little bit more complicated to use, and you need an infrastructure, a cell culture infrastructure, in order to be able to manage them and bring them into your research. Now, for most researchers that sounds quite trivial, but for others in an academic setting or in small biotechs, even setting those up can be difficult. As we layer on the complexity, as we build from simple two-dimensional cell systems to complex co-culture, three-dimensional systems, perhaps microfluidic systems, of course the infrastructures or the levels of investment in the tools that are required for cell culture becomes greater and harder to attain. Of course then, in terms of the reason why we want to use the cells and generating that transition to the clinic, the validation of our models, of course there's a challenge around trying to make sure that the cells you're working with are as close to that disease model as you want them to be. So characterizing those models, understanding them, understanding really what they're telling you. I think research and innovation and LRIG actually as a community really help scientists, pharmaceutical scientists, scientists from biotech companies, academic scientists. It helps us all come together and discuss all of the issues around cells, all of the issues around drug discovery. Ian Tomlinson this morning in the keynote at the conference here talked about those models changing, the interactions between those groups changing, and actually that we're really just scientists. LRIG, research and innovation, is a mechanism of bringing scientists together. The major challenges facing drug discovery, I think one of the reasons we've said we're interested in cell models is that transition to the clinic and representation of patient and disease models. I think that's one aspect of the challenges that the industry does face. But for me, actually, one of the biggest challenges, I think, is making the right choices as we go through drug discovery. If you think about success of drug discovery, one measure of success is how many new chemical entities come to the market. And looking at those statistics which are available from the FDA, looking at those, the last 5 years is actually the second most successful period in pharmaceuticals ever. So you have to look at that and remember that there are lots of opportunities. There are, and actually we are getting better at bringing those forward to the market as a community across drug discovery. So for me, it's now about making the right choices. Which projects to invest in, which to bring forward through, and in my line of work, pharmaceuticals, and which to accelerate through to the clinic to help patients.